Hi, this is Patricia Kopko from the University of California, San Diego, and this is the Blood Bank Guy Essentials podcast.
Hi, everyone. Welcome back to Blood Bank Guy Essentials, the podcast with just one job to help you learn the essentials of transfusion medicine. My name is Joe Chaffin, and I am your host. Today's episode is a discussion with my friend, Dr. Pat Kopko from the University of California, San Diego, about what we see and about maybe what you should consider in the brand new FDA platelet bacteria guidance that just came out at the end of September 2019. But before we get to that, you should know that this is not a continuing education episode. You can find other episodes where physicians and laboratorians can get those free CE credits at bbguy.org/podcast. Look for episodes there that end in the letters CE. You can also find those CE episodes at wileyhealthlearning.com/transfusion news. The continuing education episodes at Wiley Health Learning are brought to you by transfusionnews.com, and Transfusion News is brought to you by Biorad, who has no editorial input into this podcast. Many of you are aware that over the past several years, the United States FDA has looked very carefully at what they should do in terms of trying to help blood centers and transfusion services reduce the risk of a vulnerable patient getting bacteria in a platelet transfusion. You may think that's not necessarily a big deal, especially if you're starting out, but all you have to do is look at the FDA transfusion fatality reports every year. You'll see patients who have fatal transfusion reactions from sepsis resulting from bacteria in platelets. And also recently, earlier this year in 2019, in the Morbidity and Mortality Weekly Report, there was an episode or multiple episodes of some significant outcomes as a result of platelet bacterial contamination. It's a big deal. And FDA is aware of that. And I think all of us are aware of that. And so I think I can speak for myself and say I was very excited to see the final guidance come out from the FDA on September 30, 2019. And this guidance has a very long name. You can find this guidance on the show page for this site, or the link to it anyway, at bbguy.org/076. I would really urge you to go and get it right now, in fact, if you can, because following along, I think, will help you get more out of this discussion. I want to be clear, this discussion that Pat and I are going to have today is not a, hey, here's what you have to do as a result of what's in the guidance. What's really important to understand is this guidance gives you a lot of different options, and what may work for one hospital or one facility or one blood center may not work for yours. So there really is no one-size-fits-all strategy, but I think right now the most important thing is to try and understand what exactly is in the guidance, and that's what we're trying to do. We're trying to give you help you lay the groundwork so that you can have discussions with your blood center or with your transfusion service to try and figure out what's best for you. And my friend Dr. Pat Kopko is well qualified to have that discussion. She is a graduate of the Loma Linda University School of Medicine, my alma mater. She did her residency in anatomic and clinical pathology at Loma Linda and a transfusion medicine fellowship at Cedars-Sinai Medical Center in Los Angeles. Pat is a professor of pathology at the University of California, San Diego, where she is the director of transfusion medicine, as well as the associate director of the Immunogenetics and Transplant Laboratory. Pat is awesome. She's my friend, and I'm very excited to share this discussion regarding the new FDA platelet bacterial guidance with you. Pat, my friend, welcome back to the Blood Bank Guy Essentials podcast.
It's great to be back, Joe.
So, Pat, we've got a guidance. Holy cow, we've got a final guidance from the FDA, bacterial risk control strategies for blood collection establishments and transfusion services to enhance the safety and availability of platelets for transfusion. Hooray! Issued just a few days ago from when we're recording this on September 30, 2019. This has been quite an odyssey to get here, hasn't it, Pat? I mean, I know you've been following this, as have I. It's been quite a up and down scenario. What's your perspective on that?
My perspective is that it's been a long time coming. As you know, I really believe in transfusion safety. And I think it's important to consider patient safety. And I'm glad that this document has been published finally, because it's been a long odyssey, and I think we know that we could do more.
That really was my first question to you. For people out there that may be wondering about this, why the heck do we even need a guidance to discuss bacterial risk control? What's your take on that?
My take on that is that despite everything we've done, and let's make no mistakes about this, the transfusion industry has done an incredible job of decreasing the risk of bacterial contamination of platelets. For example, the diversion pouch, changing the arm scrub, culturing the products. We've made a significant impact on the risk of bacterial contamination of platelets, but we are not at the point where there is zero risk. I'm not sure we'll ever get to the point of the zero risk, but I think that we can further decrease the risk with some additional measures. And I think that really was pointed out by that, I think it was an MMWR publication a few months ago about the residual bacteria and septic transfusion reactions from platelets.
Yeah. And as I recall, Pat, that was, again, not to point fingers at anything or any technology, but those infections eluded even some of the things that we're going to talk about today in terms of what's in the guidance and what you've already implemented in San Diego, right?
Absolutely.
Yeah. Scary stuff. Well, I mean, the one thing that I have heard often, as you know, Pat, I work in a blood center, have worked in transfusion services for most of my career. And as part of this educational service that I provide with Blood Bank Guy, I get a lot of questions from a lot of places. One of the things that people ask me about, people say, well, isn't this just a blood center thing? Isn't this just stuff that blood centers have to do? Why do I have to care about it in a hospital?
The first reason you have to care is because we should all be concerned about decreasing the risk of bacterial contamination of platelets. So that's the first reason. The second reason is currently there is no one-size-fits-all approach to reducing the risk of bacterial contamination of platelets. And you can see that in the guidance. There are so many different options, and you have to figure out what works. If you're in a transfusion service, you have to figure out what works for you, but it also has to work for your blood center. You have to work with your blood center on this. It is not possible to impose a one-size-fits-all across the country right now, and it is also not in anyone's best interest for anybody to say, well, I must have this and only this.
That's huge. I could not agree more with that last thing that you said, Pat. It's so important as we move forward that people understand, and we're going to hear more about this in the second half of this interview in terms of how successful you guys were in implementing one particular strategy. But you're 100% right. The flexibility is going to be required, figuring out how to meet your needs with what will most likely be some combination of these strategies. I'm right there with you. I think hospitals and blood centers have to work together to figure all this out. And with that said, Pat, I want to let everyone know that our intent here, there are two things that are important for people to remember. Number one, what we're talking about is an FDA guidance that has just been finalized, as I mentioned. So obviously, those of you that are outside of the United States, you're just kind of looking over our shoulders on this one. This really does apply just to the United States, though there are lessons to be learned elsewhere as well. But different countries have different strategies and requirements. So this is a US thing, number one. Number two, what we're going to do for the next 20 minutes or so is intended to be kind of an essential guide to what we see right now in this guidance. And this is not an endorsement of any strategy. It's not meant to be regulatory advice. It's just a couple of friends, a couple of transfusion medicine docs talking through this and kind of pointing out the stuff that we see. So I think both of those are important for our audience to keep in mind as we go through this. So Pat, you ready to hit this thing, or do you have any further thoughts before we start?
No, I think we can get going.
Rock and roll. All right. So this guidance, Pat, I think, just to set the stage, there have been, I can't even remember how many draft guidances that preceded this. I know the most recent one was in December 2018. But I don't know, what was it? 2, 3, 14? I don't remember how many drafts, but it seemed like a lot, didn't it?
There certainly were at least 2 draft guidances, if I'm remembering correctly.
Well, see, that's why I have you on the podcast, Pat, because you're my voice of reality. I'm the goofball and you're the voice of reality. So
I don't know about that, Joe.
This is our relationship in a nutshell, Pat. I'm the goofball. You're the mature one. Okay. Anyway, so the guidance, which everyone will be linked on the show page for this episode, bbguide.org/076. So I believe everyone in U.S. transfusion medicine should get a copy of this guidance if you have not already. Like all guidances, it comes with the standard FDA stuff at the beginning that says you're not required to do any of this, but you kind of are. The guidance talks about several different strategies. I don't want to take the time to go through all the preamble stuff because everybody can read that. But Pat, let's just get to the heart of this. How, from your perspective, how would you divide the strategies that FDA has suggested in this guidance?
So if you're taking the big picture, they have 2 types of strategies from the perspective of, is it a one-step thing or is it a 2-step thing? And the 1-step ones are that you can do large volume delayed sampling or pathogen reduction and you're done. That's all you have to do. And so if you look at those, conceivably, all of those could be done exclusively at the blood center. But there's a problem with one of them. The one that gets you to 7 days, I believe there is nothing currently approved in that pathway.
You are not wrong. That is absolutely correct. The hope is that there will be something soon. Obviously, I don't have any inside information on that, but that's the hope that will be the case. But let's, before we get into that nitty gritty, Pat, I want to make sure that people are clear. We say large volume delayed sampling. Let's kind of at least try and define that. First, what is it? And second, how is that different from what people are doing now?
Well, for some blood centers, it's actually not different than what they're doing now. Mm-hmm. Because when we first started doing this, most blood centers started doing the culture at 24 hours and a standard volume of, if I'm remembering correctly, since I'm not in a donor center anymore, about 8 mils.
Correct.
The large volume delayed sampling is a larger volume. And so, yes. So if you look on pages 4 and 5 of the guidance document, depending on whether they're talking about 36 hours or 48 hours, it is using a sample volume of at least 16 milliliters inoculated evenly into aerobic and anaerobic culture media. This also differs from when we first started this. Many blood centers were just inoculating an aerobic bottle and were inoculating at least 24 hours. This is a change from that, but I think there are blood centers already doing the 16 mils, and there are also blood centers already doing the 36 hours.
I think that's correct. That's my perspective as well. And further, the aerobic and anaerobic thing has been implemented by many blood centers, including some large ones. It's been, I think there was a survey a few years ago, Pat, as I recall, that that looked at what the most common strategy was. And I think, and this was, I can't remember exactly when it was, but this was a few years ago. And most commonly people were doing, as you said, 8 mLs with a 24-hour hold. And everyone, just so you're clear on that, the idea is to give the bugs a chance to grow. You don't want to sample the bag as soon as you collect it. So at least 24 hours later, collect that sample, 8 mLs, put it in aerobic media. That was the most common. But now this change, as you said, there are many places that are already doing 16 mLs and are already doing aerobic and anaerobic. It's just not something that had been formalized as a requirement. So with those two strategies that you mentioned, the LVDS, large volume delayed sampling, it's that extension to 36 hours to give the bugs a chance to grow, basically, right? Isn't that what we're talking about to enhance the safety?
100%. It gives the bacteria time to lift off so that you can detect them.
So with both of those, we're talking LVDS at 36 hours or more after collection, then LVDS at 48 hours after collection. What's the difference between the two in terms of, as you said, one of them is not approved yet. LVDS 48 is not approved yet. But assuming once everything does get approved, and we're hopeful that it would, what does that change in terms of the shelf life of the product between those two?
So they're very similar, except if you wait the extra 12 hours to perform your sampling, it extends the expiration date of the product to 7 days from 5 days.
So you lose a little on the front end to get extra time on the back end, in other words.
You lose 12 hours on the front end to gain 48 hours on the back end.
It seems a reasonable trade. Yeah. Okay. So with those two single step strategies, LVDS, that may involve some changes for blood centers. And again, everyone, please read the guidance yourself. We're not endorsing, and but we want to point out what some of the strengths of this, of each strategy might be. As you said, those are blood center things. And the potential is that a transfusion service could get a product that could either go to 5 days or potentially to 7 days once that's approved without them having to do anything else, right?
Yes.
Well, what about the third strategy? Not necessarily third in terms of importance, but third in terms of the order that it's listed in the guidance, and that's pathogen reduction. I know you have a lot of perspective on that. We're going to talk in the second half of this interview about that. But what about pathogen reduction in terms of shelf life for the product?
Currently, the one approved pathogen reduction process for platelets is approved for 5 days. And so what we will see in the guidance document, it says specifically it does not give an expiration date for the product. It says per device instructions for use.
Obviously, more to come on pathogen reduction in just a little bit, but I guess we should summarize the single step strategies. So there are 3 in the guidance. LVDS, large volume delayed sampling, at no sooner than 36 hours, that you do aerobic and anaerobic culturing. It gives you a 5-day product. The same process, but waiting until at least 48 hours. Again, aerobic and anaerobic, it gets you to 7 days. Oh, and one other thing, Pat, we should point out, and this is more for blood center nerds like me and like you used to be. Now you're cool. But that one other change is that you're required to sample each part of a split. And that is potentially kind of a big deal for us in blood center world. Put your old blood center hat on for a second, and you can see if you've got to sample each split. And that's like for a triple, for example, you're talking 6 bottles at least, and 32, 48 mLs of sample that you're losing. That can be significant.
That's a lot more cultures than when we originally started. If you think back when we originally started, we were doing 1 culture on the entire bag.
Right. So those of you that are listening that are like, triples, what's he talking about? The thumbnail. Real quick, for apheresis platelet collections, most donors we're able to collect enough to split the product into 2 doses. For those of you taking tests, each of them more than 3 times 10 to the 11th platelets. Sometimes we can get 3 doses. Rarely we can even get 4. But what we're saying is every one of those individually split products has to be cultured, and that's a big change. That's a big difference. So Pat, before we move on to those 2-step strategies, anything else that you want to point out about those single-step strategies?
Yes, there is. For anyone listening along and has the guidance document in front of them, what you just said about you have to sample each of the splits is on page 5 at the top under heading number 2. If the apheresis product will be split, each split unit should be sampled. I think that people might not grasp because we haven't been doing this all along. And it's what you said about the triples, that if you're now collecting a triple, you have to do 6 cultures on that where we used to do just 1.
That's a big deal. Big thing. Okay. Well, Pat, 2-step strategies. So the way I think about 2-step strategies, and you tell me if I'm way off on this, but the way I'm looking at it is that what the 2-step strategies do is it gives you a way that if a blood center, say for example, wanted to stay with close to the status quo of what they've done before, taking that primary culture at 24 hours with some caveats. You still have to do aerobic and anaerobic in this protocol. Then they could theoretically do that. But the problem is that doing that leaves more work to be done. Is that how you see it too, Pat?
That is true. And at first I was wondering why the FDA even put this in the document, why anyone would even do that. But as I've been thinking about it, if you need a platelet fairly quickly, so if you need an HLA matched platelet or a cross matched platelet, and you don't want to wait an extra 12 hours, this is a good option to use.
You know, I hadn't thought about that, Pat. That is a great thought. You're right. That is one possibility. So let's back up for just a second. So 2-step strategies. Obviously, there's got to be a step 1 and a step 2. Why don't I shut up for a second and let you summarize the 2 ways that you can do the first step of that testing?
So for the first step 1, you can either do a primary culture no sooner than 24 hours after collection, or a large volume delayed sampling no sooner than 36 hours after collection. And again,
wait, that sounds familiar.
Yes.
Is that the same LVDS 36 that we just talked about?
It is the same LVDS 36 that we just talked about.
Ah, okay.
And at first, as you know, because we've been texting back and forth about this, that confused me senseless. I didn't get it.
and it's so rare for that to happen, Pat. I have to tell you that I kind of enjoyed it. I was like, wait, something confused Pat. That's a miracle. That just doesn't occur. So thank you for letting the 12-year-old in me be pleased with your confusion for a moment.
Well, I just didn't get how you would do the large volume delayed sampling as a 1-step, but then do the same exact thing as a 2-step.
Okay, so what did you figure out, my friend?
Well, I didn't figure it out because you explained it to me.
Whatever.
And so what we have figured out is that if you're going to do the large volume delayed sampling and you want to extend the expiration date of the product to 7 days, you have to do a 2-step process.
So you could do that LVDS at 36 hours and have a 5-day product without doing anything else. But if you want to go to day 6 or day 7, then you've got more work to do.
Yes.
And I want to point something out about the primary culture no sooner than 24 hours, because I think this is really interesting. And this is on the top of page 7, you guys following along, that if you're doing that primary culture 24 hours, it says you can actually label that it's going to come to you if you're a transfusion service labeled with a 5-day expiration. but you can't actually transfuse it to 5 days. The guidance says to assure that the risk of bacterial contamination is adequately controlled for transfusion after day 3, you have to do some sort of secondary testing that we're going to describe in just a minute. To my knowledge, I'm not aware, Pat, correct me if I'm wrong, I'm not aware of any product ever that has been it has a 5-day expiration label, but you can't use it to 5 days. Are you aware of anything else that's been like that?
No, and that's what got me thinking about why would they even do this? This makes no sense. And that's when I thought of, for all the times you need platelets quickly, you can still do the culture at 24 hours and transfuse the product up to 3 days.
So that's step 1, guys. Step 1 gets you either with the first option, functionally a 3-day platelet product, which is, you and I both know, Pat, blood centers really can't turn around platelets much faster than, if you're lucky, a day and a half, but between a day and a half and 2 days after collection. So you're talking like a little over a day on your shelf product, sadly. Or if you do the large volume delayed LVDS 36-hour, you get a 5-day product. Now, let's talk through the step 2 processes, which are really fairly straightforward to my eye anyway, Pat. And if they're straightforward to me, I'm sure they're straightforward to you. Tell us what people have to do to get either to 5 days using option 1 or beyond 5 days.
So there's 2 ways you can go. You can either use a secondary culture or secondary rapid testing. If you're using a secondary culture and you've opted for testing the product no sooner than 24 hours after it was collected, then you have to do a secondary culture no sooner than 3 days after it was collected, and that extends the expiration date. Actually, it maintains it at 5 days. So it's that thing you were talking about, that it's labeled as 5 days, but you can only use it to 3 days unless you do an additional culture. So that's the first culture. The second culture option is that at no sooner than 4 days, you can do another culture, and you have to, for both of these, you have to culture each split. And if you do that, that can extend the expiration date to 7 days. And so I think that would apply to the large volume delayed sampling.
What about the last option, the last second step test?
So the other second step is secondary rapid testing. And that needs to be done per device instructions. And you can extend the life of the product up to 7 days if you follow the device instructions provided by the manufacturer.
Yeah, and we're being slightly vague on that for good reason. I mean, the guidance is vague. The guidance pretty much says do it according to the package insert. I think most everybody knows what we're talking about here. The secondary rapid test that's typically done. I've joked about this test before and called it the platelet pregnancy test because that's kind of what it looks like. but it's significantly more complex to do than a platelet pregnancy test, right?
Yes, the platelet pregnancy test is significantly more complicated to do than the regular pregnancy test. And with the platelet pregnancy test, you never want it to be positive.
That is a fair statement. That is true. So with that, oh my gosh, I don't know how I'm going to recover from that one. So with that test, again, and we're not endorsing, criticizing, or anything, but the bottom line with that test is that when it's done, according to my interpretation of their package insert, when it's done, it gives you 24 hours from the time that the test is done to extend that platelet shelf life. Is that your understanding as well, Pat?
That is my understanding as well.
people can handle this different ways. I know of facilities that are using this test currently, even prior to the guidance being finalized. We should be clear, that test is currently approved for extension to 7 days right now. Even before this guidance came out, people could do that and extend the product to 7 days. Again, as long as you're testing beyond 5 days within 24 hours of the time the product is transfused.
Absolutely. But that's the same with much of this guidance document. Much of this guidance document and many of the things in it, blood centers and hospitals are already doing.
It formalizes a lot of it and, adds some new requirements to do, for example, anaerobic testing, as we've talked about, but many places were already doing that anyway. So, Pat, before we get to your overall impressions of how this does what we were hoping it would do, there is one thing that I think is super important for hospitals in particular to be aware of. And I'm just going to throw it to you to talk about perhaps the new regulatory stuff that could come from implementing those two-step processes. What can you tell us about that?
I think the most important thing for transfusion services to know is that if you intend to use a two-step process and you will be doing the second step in your transfusion service, you have to register with the FDA as an entity that manufactures blood products, if you have not already done so. And that is on page 13 of the guidance document. And I just want to read a brief passage. It's the implementation of a bacterial detection device that is used to relabel a platelet product with a 6 or 7 day expiration date, thereby extending the dating of the platelet product, is a manufacturing procedure requiring registration and blood product listing as described in 21 CFR 607.3D. Transfusion services that implement secondary testing on platelets with a 5-day expiration date are not required to register and list because they are not extending the dating period of platelets. So, in summary, if you are going to do that second step and you're going to extend the expiration date of your platelet to 6 or 7 days based on testing that you are doing in your transfusion service or in your microbiology lab or anywhere else in your facility, you need to register with the Food and Drug Administration as a blood product manufacturer if you have not already done so.
That is something that, quite frankly, some people will view that as a major deal, I think. Other people might not see it as that massive a situation, but it is definitely something that hospitals need to consider as you're moving forward with this process. As we said before, everybody's going to have to look at this and see what works best for them, what works best within their way of practicing. I think maybe, and Pat, you and I have talked about this before offline, that a rural hospital might have a different answer than a large hospital, a large academic hospital that's in the middle of a city. The answers may be different where you are, but that's definitely something. What Pat just pointed out is so important to consider, that it does change your regulatory status. If you're going to try to get to 6 or 7 days, it changes things for you. Let me be clear. If you're going to try and get to 6 to 7 days based on something you are doing at your facility, it changes things for you.
Yes. And so it depends on who you are. Our hospitals are already registered with the FDA because we do manufacturing already. But if you are a smaller hospital, you may not do any manufacturing in your transfusion service. And if you decide to extend expiration date using point of issue testing or culture, then you have to register with the FDA, which means that you will be inspected by the FDA approximately every other year.
So it's just something to add to the calculus, right? It's something that over the next 18 months, everybody's going to have to decide what works best for them and what scenario fits best into their strategies, as I said. Okay, so Pat, we've kind of talked about the main points from our perspective in this guidance. There is other stuff in there. We haven't really talked about how to deal with this for whole blood derived platelets and pools of whole blood derived platelets. All that is in the guidance. I would refer you to it. We're focusing on, the 85-90% of platelet transfusions in the United States that are apheresis. So that's been our focus. And again, I think we should just summarize one more time so people can follow along. You've got a 2-step strategy and a single-step strategy. The single-step strategy involves taking large volume delayed sampling cultures either at 36 hours, which gives you 5 days of shelf life, or 48 hours, which once it's approved would give you 7 days of shelf life. Or you can use pathogen reduction, which currently will give you a 5-day shelf life. And hopefully that will change at some point. Or you have the 2-step process whereby you have step 1, a culture at 24 hours that gives you functionally 3 days of shelf life, or that same large volume delayed at 36 that gives you 5 days, followed by secondary cultures done either at day 3 to get you to 5 days, or day 4 to get you to 7 days, or the secondary rapid testing, the point of issue approved safety measure test that in theory could, well, actually more than just in theory, could get you up to 7 days of storage. How'd I do with that, Pat? Does that sound about right?
That sounds like a good summary to me.
You can find references for all this on the show page. Please check it out, bbguide.org/076. But I think, Pat, I want to spend just a few more minutes with you before we get into the other stuff that we want to talk about, which is hugely important. I want to spend just a few minutes with you talking about what hospitals should do now. And I probably should have brought this out at the beginning. So the FDA has put out a guidance. There is a final guidance out there. That sounds ominous. That sounds scary. What does it functionally mean? What do we need to do today?
Today, you need to read the document.
Yes.
That's where you start. Read the whole document. And one of the things you will discover is that the FDA has given us 18 months to completely implement this. So this doesn't have to be done today, but you really need to get working on it. And then the second thing transfusion services need to do is that they need to talk to their blood services provider and find out what their plans are. In fact, some of you will find out that you're already getting platelets that are collected and tested under the single-step method, because there are blood centers already doing the large volume delayed sampling at greater than or equal to 36 hours. Now, they're probably not testing every split, but you should not be surprised if your blood center is already doing a lot of this, but just has to implement a few more things. But you're not going to know unless you talk to them.
I would tell you my perspective, Pat, and believe me, I haven't done surveys of every blood center in the United States, not by any stretch. My suspicion, as you said, is that you're definitely going to find places that are doing aerobic and anaerobic. I think you're definitely going to find some places, I think, doing 36. I don't know. No one to my knowledge is doing 48. It's the big thing is culturing each split that I think is very much in the minority today in the United States. At least that's my perspective. I could be wrong about that, but I don't think there's a ton of places doing that.
I think you're right about that.
And quite frankly, there are other organizations that are doing some even larger volume sampling than some of this that's described. Anyway, there's a lot of strategies out there that are being used. I think that this kind of funnels things more in specific directions. And as you said, you do not know, or you may not know until you ask what's going on now and what the blood center's plans are. But don't be surprised if you call your blood center and they say, we're still figuring it out too, because that's possible as well.
Absolutely. Before
we leave this, Pat, I just kind of want to get your overall take and circle back to what you said at the beginning. this is the editorial part of this podcast, and far be it from either of us to criticize the FDA, and that's not the intent of this. But do you think that this moves the ball down the field, to use a football analogy, in terms of what you said at the beginning? And I completely agree with that. Our goal should be to keep our patients as safe as possible. Do you think that the steps outlined in this guidance move the ball in that direction?
I think they do. As we just discussed, you're going to find that a number of blood centers are doing some of this already, that the blood centers have moved the ball down the field. But I think there are very few blood centers that are doing the large volume delayed sampling and 16 mils per split and anaerobic and aerobic cultures. So I do think it moves the ball down the field. And then I think there are also very few sites that are using either a secondary culture or secondary rapid testing to extend the shelf life of the platelet. And there are also some, and a growing number, using pathogen reduction. But this formalizes that you have to do one of these.
Again, everyone, please read the guidance yourself. Everything Pat and I said is not any official position either of UC San Diego or of Lifestream or of anybody. It's our opinion based on what we're seeing. But check the guidance out. We're going to actually issue this as a standalone episode, just this discussion on the FDA guidance. And part 2 of this discussion will be a separate episode where Pat and I talk about her implementation of one of these strategies at UC San Diego prior to this guidance even coming out. So, Pat, I will look forward to talking to you next time about your program at UCSD.
Me too, Joe.
Hi, it's Joe with just a couple of quick closing thoughts. As I mentioned, the next episode of this podcast, episode 077CE, will be the continuation of my discussion with Dr. Kopko. Pat and I go into the details, actually, about how she has implemented nearly completely one of the options that we described in the interview today that was mentioned in the platelet bacterial guidance from the FDA. Pat and her team have published on this, and they have a paper that is in early view in Transfusion that you're welcome to check out before that episode. And I don't know if I mentioned this, but that episode will be continuing education eligible. So I hope you come back and listen. As always, you can hear past and future episodes of this podcast directly on the website at bbguy.org, or you can go to Apple Podcasts or Google Play or Spotify, really, wherever you find your podcasts. I do want to especially thank those of you that have left ratings and reviews on Apple Podcasts. I see all of those, and I'm very, very appreciative of the kind words. I have been away for a while. Some of you have noticed, and thank you for writing me with your concerns. I'm fine. It's been a tough summer. There were some things going on that I'm not quite ready to share that have been very difficult for me personally, but all is well now. And thank you again for your concern and your kind words and your letters. So the next episode, as I said, episode 077CE, will be out very soon. I'm looking forward to sharing it with you. But until that day, my friends, as always, I hope that you smile and have fun. And above all, never, ever stop learning. Thank you so much for listening. I'll catch you next time on the Blood Bank Guy Essentials podcast.